Welcome back to the deep end. We are diving back into the microscopic war zone that is, well, the human immune system.
It really is a war zone. But, up until now, we've really only been talking about a very specific kind of warfare.
Right. If you've been with us for the previous deep dives, specifically looking at the innate immune system, you'll know we've described it as the carpet bombing strategy.
That's a great way to put it.
It's the first responders. Right. It's the macrophage seeing a bacterium and just saying, that looks vaguely bad, let's burn the whole neighborhood down.
Exactly. It's fast, it's angry, but it is definitely not subtle.
Not at all.
The innate system, it relies on what we call pattern recognition. It sees a flagellum, a piece of a bacterial cell wall, and it just fires. It really doesn't care if it's E. coli or Salmonella.
It just knows it's a bacteria.
It just knows it's a bacterium.
Yeah.
But today, we are shifting gears entirely. We are moving from that blunt instrument to the scalpel. We are talking about adaptive immunity.
The special forces, the snipers, the intelligence agency of the body.
All of the above. This is Micro-9, antigen-specific immune responses. We are basing this deep dive primarily on the structural spine of Chapter 9 of Murray's Medical Microbiology.
And we're also pulling in some heavy clinical artillery from Chapter 8 of Girouette's, Melnick, and Edelberg's.
We are. It's a great supplement for the clinical side of things.
And the mission today feels pretty epic. We are mapping the entire journey from a naive recruit, a lymphocyte that knows absolutely nothing, to a specialized weapon of war. One capable of remembering a specific enemy for decades.
And that's the key word right there, remembering.
I wanted to start with that, because Murray, he outlines 3 core principles for adaptive immunity: specificity, memory, and inducibility.
Those are the absolute pillars of the system. Without them, it just doesn't work.
So let's unpack them, because they really explain why this whole system even exists. Let's start with specificity.
Right. So unlike the innate system, which, paints with a very broad brush.
The carpet bombing.
The adaptive system is completely laser-focused. Specificity means we are not just recognizing bacteria. We are distinguishing a single specific protein sequence on one particular strain of a specific virus.
It's a difference between recognizing a military uniform and recognizing a specific soldier's fingerprint.
That's the perfect analogy, and that specificity leads directly into the next pillar, which is memory.
This feels like the holy grail of immunology.
It is. It truly is. Once the adaptive system meets an enemy, it keeps a file on them. It creates these memory B and T cells, and if that same enemy shows up again, maybe 10 years later...
The response is totally different.
Totally different. It's not that laggy, slow startup we see the first time. It is instantaneous, it's massive, and it usually wipes out the pathogen before you even feel a single symptom.
Which is the entire biological basis for every vaccine ever made.
Exactly. That's all a vaccine is. We are hacking the memory system to give it that most wanted poster ahead of time.
Okay, and the third one, inducibility. This one always sounds a bit ominous to me, like a sleeper agent.
That's actually a perfect analogy. It is a sleeper agent.
Yeah.
You have to realize the adaptive immune system is incredibly powerful, but it's also incredibly expensive, biologically speaking.
Consumes a lot of energy.
Massive amounts of energy and resources to build these millions of specific cells. You simply cannot have them running at full capacity all the time.
You just burn out.
You'd starve, literally. So the system sits dormant. Murray calls it a sleeping giant, and that's what it is. It waits until it is specifically triggered or induced by an antigen.
And only then does it wake up.
Only then. It wakes up, it proliferates wildly in a process called clonal expansion, and it goes to war.
So today we're going to trace that induction. We're going to look at how the body distinguishes a harmless protein from a deadly one, how T cells go to university in the thymus, and how they coordinate with B cells to launch this precision-guided attack.
And just a quick note for the medical students tuning in, this is extremely high-yield territory. We're going to be flagging the mechanisms that show up on board exams, the why behind clinical presentations, and specifically the exam traps that trip people up.
Absolutely. So let's start with the basics. The language this war is fought in. We have to define our terms. Immunogens versus antigens. I feel like these get used interchangeably all the time, even in clinical settings.
They do, and in casual conversation, it's mostly fine. But in the strict language of immunology, and certainly on your exams, there is a very important difference.
Okay.
An antigen is strictly defined as anything that can bind to a specific receptor. That could be an antibody, it could be a T cell receptor, it just has to fit the lock.
But just because it fits the lock doesn't mean it turns the key and opens the door.
Precisely. That's where the term immunogen comes in. An immunogen is a type of antigen that actually triggers an immune response. It induces that activation we just talked about.
So if we're being logical about it,
Yeah,
all immunogens are antigens, but not all antigens are immunogens.
exactly. You can have a molecule that your antibodies can see and bind to, but it doesn't cause the immune system to wake up and fight. It's inert.
And this brings us to a massively important concept for pharmacology and pathology, the hapten.
Haptens, yes. These are the classic example of an antigen that is not an immunogen on its own.
They're the incomplete immunogens.
Right. The word itself comes from the Greek hapten, which means to fasten, and that's what they do. Haptens are very small molecules, usually simple chemical structures. They're just too tiny to trigger an immune response by themselves.
Why is that? What's the mechanism?
Well, to activate a B cell, for instance, you generally need to cross-link multiple B cell receptors on the surface at the same time. A tiny little hapten can only bind to one. It can't bridge the gap. So it's essentially invisible to the surveillance system.
Which makes sense from an evolutionary standpoint, right? You don't want your immune system freaking out over every tiny amino acid or simple sugar floating in your blood.
That would be absolute chaos. It would be constant, low-level, pointless inflammation. So the system ignores small, simple molecules. But haptens can become very dangerous if they find a partner.
The carrier molecule.
The carrier protein, exactly. If a hapten binds chemically to a much larger protein, and usually it's one of your own body proteins, like albumin in the blood or a protein on a cell surface.
It creates a new complex.
A new, much larger complex. And the immune system looks at this and says, whoa, that protein looks wrong. It's been modified.
It's the concept of modified self. It's no longer just you.
Yes. The immune system doesn't recognize the carrier as foreign, but it sees the whole hapten carrier package as something new and threatening. And the classic high-yield clinical example here is penicillin.
I was just reading about this in jail. Penicillin itself, the drug molecule, is tiny. It's a hapten. So technically you shouldn't be allergic to it.
And the vast majority of people are not. But penicillin has a chemically reactive component, the beta-lactam ring. In some susceptible individuals, that ring can break open and spontaneously form a covalent bond with host proteins.
So it just sticks itself onto your own cells.
Right. It forms a hapten carrier complex. So when someone goes into anaphylaxis from a dose of penicillin, it's not the drug itself they're reacting to, it's their own proteins that have been decorated by the drug.
That's incredible. So the immune system is actually attacking itself just because it's wearing this little penicillin hat.
That's a great way to think about it. The B cells will produce antibodies that recognize the penicillin part, the haptoligand, but the T cells, which are required for a big response, will recognize a piece of the carrier protein. This cooperation is what triggers the massive, dangerous allergic response.
Okay, so speaking of triggers, Murray makes this really big distinction between two types of antigens based on how they activate B cells, T-independent versus T-dependent. This feels like a major fork-in-the-road moment for the immune response.
It is the most critical fork in the road for antibody production. It determines everything that follows.
Let's start with T-independent. The name kind of gives it away.
It does. T-independent antigens are generally large, highly repetitive structures. Think of the things on the outside of a bug, the armor.
So bacterial capsules made of polysaccharides or flagellin from the flagella, or lipopolysaccharide, LPS.
Yeah, exactly. Because these molecules are so big and possess all these identical repeating subunits, like a long chain of the same sugar over and over again, they can physically cross-link multiple immunoglobulin receptors on a B cell surface at the exact same time.
Okay, so imagine a B cell that's just covered in these Y-shaped antibody receptors. A long bacterial capsule comes along and lays across them like a piece of Velcro, grabbing 10 or 20 receptors at once.
That's a perfect visual. That Velcro effect sends such a strong, overwhelming signal into the B cell that it gets excited and starts firing without waiting for a second opinion.
It doesn't need to check in with the T cell.
It activates independently of T cell help, hence T-independent.
It sounds efficient. I don't need a commander, I'm just going to shoot.
It is fast. It's a fantastic early defense mechanism, but there's a massive catch. There are actually two big catches.
Okay.
Because it bypasses the sophisticated help signals from a T cell, you get a very limited, very primitive response. First, you pretty much only get one type of antibody, IgM.
And we'll talk about all the antibody types later, but IgM is kind of the basic first responder model.
It is. But the second and most critical catch for clinical practice is this. With T-independent antigens, you do not generate immunological memory.
No memory. That seems like a huge design flaw. If I get infected with a capsule-bearing bacteria like strep pneumo, I definitely want to remember it for next time.
You do. But the B cell needs specific signals from the T cell to build those memory files, to undergo the maturation process that leads to a long-lived memory cell. T-independent activation just doesn't provide those signals.
And this has massive clinical implications, right? Particularly for vaccines in children.
This is the entire why behind the conjugate vaccine schedule. It's a huge point in both Murray and Jowett's. Infants, especially children under 2 years old, have very immature immune systems. And they are particularly terrible at handling these T-independent polysaccharide antigens.
So if you give a baby a plain polysaccharide vaccine, like the old pneumococcal vaccine,
Nothing happens. They won't make protective antibodies. Their B cells just kind of stare at it. The response is either non-existent or very weak and short-lived.
so how do we fix that? Because we absolutely need to protect babies from things like pneumococcus and Haemophilus influenza type B, there's both encapsulated bacteria.
We get clever. We use the hapten carrier principle we just talked about, but in reverse.
Okay.
We take that sugar capital, which is a T-independent antigen, and we chemically attach it, we conjugate it, to a big immunogenic protein like tetanus toxoid, which is a T-dependent antigen.
Ah, the conjugate vaccine, like the hen vaccine or Prevnar.
Exactly. Now watch what happens. The infant's B cell has a receptor that recognizes the sugar, so it binds the sugar part of the vaccine. But because the protein is attached, it pulls the whole complex inside. It then processes the protein part and displays it on its surface to a T cell.
And the T cell knows how to respond to proteins.
The T cell says, hey, I recognize that tetanus protein. This is a real threat. And it provides all the powerful activation signals to that B cell.
That is absolutely brilliant. You're tricking the system. The B cell thinks it's fighting a sugar, but the T cell helps it because it sees a protein.
You're linking the two responses. And because you've engaged the T cell, you unlock the entire premium package of adaptive immunity.
So you get class switching to make the powerful IgG antibody, you get somatic hypermutation to make high-affinity antibodies, and most importantly,
You get long-term memory. That's why conjugate vaccines work so well in babies and provide lasting protection.
okay, so that establishes the targets of the war. Now let's talk about the soldiers, specifically the T cells. Murray describes their development as a sort of rigorous education system that takes place in the thymus. But honestly, hearing the details, it sounds less like a university and more like a scene out of the Hunger Games.
It's absolutely a brutal selection process. A pass or die boot camp is a very good way to put it. You have to realize T cells are born in the bone marrow, but they're born naked. They don't have their specific T cell receptors yet.
They're a blank slate.
A total blank slate. They travel to the thymus, this little gland sitting on top of the heart, to get their identity and to be tested.
And this is where the math just gets scary.
Yeah.
What percentage of T cells that enter the thymus actually survive to graduate?
It's shockingly low. It's somewhere around 2 to 5%.
So a 95 to 98% death rate. That seems incredibly inefficient. Why does the body waste so much energy making all these cells just to immediately kill them?
Because the cost of a bad T cell escaping into the body is catastrophic. The cost is a fatal autoimmune disease. We're generating these T cell receptors completely randomly, which we'll get to in a minute, so we are absolutely bound to make mistakes. We're going to make receptors that attack our own bodies. The thymus is the filter that catches those mistakes.
And it happens in 2 major stages, right, as they move through the anatomy of the thymus.
Yes, it's a physical journey. They start in the outer part, the cortex, and if they survive, they move to the inner part, the medulla. Each location has a different test.
Let's walk through the cortex first. This is called positive selection.
Right. In the cortex, the young T cells, we call them thymocytes at this stage, and they're double positive, meaning they express both CD4 and CD8 molecules, are given their first test. The cortical thymic epithelial cells hold out the body's ID badge.
The MHC molecule, major histocompatibility complex.
Exactly. And the question in positive selection is brutally simple. Can you read the ID badge? Can this T cell receptor that you just randomly generated interact with your own MHC molecules at all?
If the answer is no,
If the receptor is completely blind to your own signaling system, it's useless. It can't communicate. It's a dud.
well, I guess the axe.
It gets a signal to undergo apoptosis, or programmed cell death. We only keep the cells that can bind to self-MHC. This principle is called MHC restriction. Your T cells are restricted to seeing antigens only on your own MHCs.
Okay, so the 10 or 20% of cells that can actually see the ID badge survive. They move to the medulla. Step 2, negative selection.
Now the question flips entirely. We know you can read the ID badge. The new question is, do you react to what's written on the badge too strongly?
Meaning, do you recognize self-peptides?
Precisely. In the medulla, these thymocytes are shown pieces of your own body proteins, your own lung tissue, your pancreas proteins, your thyroid proteins, all presented on those MHC molecules. If a T cell binds too tightly to any of these,
That's a disaster waiting to happen. That's an autoimmune disease in the making.
that T cell is a loaded gun pointed at your own organs. So if it binds too tightly to a self-antigen, it is also given the signal to die. Apoptosis again.
So this is central tolerance, getting rid of the self-reactive cells at the source.
Exactly. You are trying to find the Goldilocks T cell, one that recognizes MHC just enough to communicate, but ignores the self-antigens presented on it. Not too hot, not too cold, just right.
Now, this part has always blown my mind. How does the thymus, this small gland in your chest, know what a pancreas protein or a retina protein or a skin protein looks like? It's not in the pancreas. How does it get samples?
That is the magic of a single gene called AIR.
AIR, autoimmune regulator.
It's one of the most incredible genes in the genome. It's a transcription factor that is expressed in the medullary thymic epithelial cells, and it allows these cells to break all normal rules of genetics.
How so?
It turns on genes that should only be expressed in the periphery. It forces the thymus cells to make a little bit of insulin, which should only be in the pancreas. It makes myelin, basic protein from the brain. It makes thyroglobulin from the thyroid. It turns the thymus into a magic mirror of the entire body's proteome.
So it creates this library of self to test all the recruits against. Here is a piece of your pancreas. Do you attack it? Yes. You're eliminated.
Exactly. If a T cell attacks the insulin protein it sees in the thymus, the AIR system ensures that T cell is destroyed long before it can ever get into the bloodstream and cause type 1 diabetes.
And if that AIR gene is broken,
That's a classic board exam pathology. It's a disease called autoimmune polyendocrine syndrome type 1, or APS1.
what happens?
The thymus fails to show those self-proteins. The autoreactive T cells that should have been killed survive negative selection, they graduate, they escape into the blood, and they just wreak havoc. They attack the patient's parathyroid glands, their adrenal glands, their skin, their gonads. It's a multi-organ autoimmune meltdown.
That is a very high-yield clinical correlation right there. Okay, so we've selected the best soldiers, but we kind of skipped a crucial step. How did we generate all these trillions of different receptors in the first place? Murray mentions we can recognize something like 10 to the power of 11 different antigens.
Or even more, an astronomical number.
But the human genome only has about 20,000 to 25,000 genes, so the math just doesn't work. We can't possibly have one gene for every single receptor.
No, we can't. The solution the body came up with is incredible. It's basically a genetic lottery, a process called VDJ recombination.
And this happens in the bone marrow for B cells and the thymus for T cells before they ever see an antigen, right?
Correct. This is all preemptive. It's the only time in biology where we intentionally break and reshuffle our own DNA.
Okay, walk us through the mechanic of that,
Yeah.
because it sounds risky.
It is, but it's a controlled chaos.
Okay.
So in the DNA that codes for the receptor, you don't have one complete gene. You have gene segments. Imagine a deck of cards split into 3 piles.
Okay.
You have a pile of variable V segment cards, a pile of diversity D segment cards, and a pile of joining J segment cards. For the T cell receptor beta chain, you might have, say, 50 different V cards, 2 D cards, and maybe 13 J cards.
So a library of parts.
Your library parts. And the cell uses a special set of enzymes, the most important being RAG1 and RAG2, recombination activating genes.
The RG enzymes.
These enzymes act like a dealer at a casino. They go into the DNA, and they randomly pick 1 V card, 1 D card, and 1 J card. They then physically cut the DNA, throw away everything in between, and splice those 3 chosen segments together.
So V3 might get stitched to D2, which gets stitched to J9.
Right. And just that combinatorial diversity alone generates thousands of possibilities. But it gets even more random. An enzyme called TDT, terminal deoxynucleotide transferase, comes in at the junctions where the segments were stitched together, and TDT just adds random nucleotides. It's not copying from a template, it's just making them up.
That's called junctional diversity.
Exactly. It's like throwing a handful of glitter into the mix. This creates a completely unique, hypervariable genetic sequence at the binding site of that specific receptor. The combination of VDJ choice and TDT additions is what generates the astronomical diversity.
But this randomness is precisely why the thymus selection process is so deadly, right? If you're building receptors randomly, you're inevitably going to build some that are perfect matches for your own self-proteins.
Precisely. VDJ recombination creates the massive arsenal. The thymus selection process provides the safety checks and ensures you don't shoot yourself.
And one more clinical tie-in.
Yeah.
If you're born without those Rh enzymes.
You can't perform VDJ recombination, so you can't make B-cell receptors or T-cell receptors.
Which means no B cells and no T cells.
You get SCID, severe combined immunodeficiency, the classic bubble boy disease, a complete lack of an adaptive immune system.
Incredible. Okay, so the T cell has its unique receptor, the TCR. It has survived the thymus boot camp. Now it's a naive T cell and it enters the circulation. But as we said earlier, T cells are, well, they're fussy. They don't just grab antigens out of the air like antibodies do.
No, not at all. T cells are effectively blind to raw floating antigen. They suffer from that MHC restriction we talked about. They will only see an antigen if it is properly processed and presented to them on a silver platter.
The MHC molecules. And Murray uses some great food metaphors here, or maybe that's just us being hungry. But let's stick with the classic structural analogies because they really help distinguish class 1 from class 2.
Let's do it. MHC class 1 is the pita pocket, and MHC class 2 is the hot dog bun.
Break that down for us. Why a pita pocket?
The peptide binding groove of an MHC class I molecule is structurally closed at both ends. It has these anchor residues that hold the peptide in place, so it can only hold very short peptides, typically about 8 to 9 amino acids long. If the peptide is too long, it literally won't fit in the pocket.
Okay, so it has to be a very specific size. And where do we find these pita pockets?
MHC class 1 is found on all nucleated cells. That's a key point. Your lung cells, your liver cells, your skin cells, your neurons, basically every cell in your body except for red blood cells.
And what are they serving in this pita pocket?
They are serving homemade dishes. We call this the endogenous pathway. Endo meaning from within.
So proteins made inside that very cell.
Right. Every single cell in your body is constantly breaking down and recycling its own internal proteins. It takes normal aging proteins, or maybe viral proteins if it's infected, or mutated tumor proteins if it's cancerous.
And it puts them through a molecular shredder.
The proteasome, exactly. The proteasome chews up these proteins into short peptides. These peptides are then transported into the endoplasmic reticulum using a specialized transporter called TA.
Transporter associated with antigen processing.
That's the one. In the ER, these peptides get loaded into the brand new MHC class I pita pockets, and the whole complex gets sent to the cell surface for display.
So it's like a continuous, real-time window into the soul of the cell. It's saying to the immune system, here is a sample of every single thing I am making inside myself right now.
Perfectly put. And the immune cell that is specialized to look in this window is the CD8 cytotoxic T cell, the killer T cell. If a CD8 cell looks at a lung cell's MHCI and sees a normal lung peptide, it moves on. If it sees a flea virus peptide, it knows that factory has been corrupted.
And its job is to kill that factory.
Eliminate it before it can release more virus.
Okay, so that's class 1, endogenous pathway, on all nucleated cells. Serves peptides to CD8 T cells. Now, what about the hot dog bun?
That's MHC class 2. The binding groove here is open at both ends. It doesn't have those restrictive anchor residues, so it can hold much longer, peptides 11, 12, even up to 20 amino acids long. They can hang out the ends, just like a hot dog in a bun.
And who has these hot dog buns? Not every cell.
No, only the professionals. We call them the professional antigen-presenting cells, or APCs.
And that's a very exclusive club.
It is. The main members are dendritic cells, macrophages, and B cells.
And they aren't serving homemade food from inside.
No, they serve takeout.
Yeah.
This is the exogenous pathway. Exo, meaning from outside. These professional APCs are the scavengers. They roam the body and eat things from the environment, bacteria, cellular debris, foreign proteins, through phagocytosis.
They digest them in lysosomes.
Right. And here's a really cool bit of molecular engineering. The MHC class 2 molecule is also made in the ER, just like class 1. But we don't want it to get clogged up with the cell's own internal junk peptides that are floating around in the ER.
Right, because its job is to display external stuff.
So it's synthesized with a parking boot on it, a placeholder protein called the invariant chain. This chain physically blocks the binding groove.
So nothing can get in.
Nothing can get in. The MHC2 molecule then travels to the lysosome where all the digested bacterial bits are. In that acidic environment, the invariant chain gets chewed up, leaving just a little piece called CLIP in the groove. Then another molecule, HLA-DM, comes and pops the CLIP fragment out, allowing the exogenous bacterial peptide to finally bind.
That is an incredibly elegant design to ensure you're only loading the right cargo.
It is. So, to summarize the function, MHC class I on a regular cell is saying, help, look what I am infected with.
Kill me.
Essentially, yes. But MHC class 2 on an APC is saying, look what I just found and ate out in the neighborhood.
Don't kill me, I'm the messenger.
Exactly. Don't kill the messenger. And the immune cell that receives this message is the CD4 helper T cell, the general.
So why the strict separation? Why have two distinct pathways?
Because it dictates the appropriate response. If a cell shows a viral peptide on MHC-I, it means that specific cell is corrupted and needs to be eliminated. You send in the CD8 assassin to kill it directly. But if a macrophage shows a bacterial peptide on MHC-T, it means there's an infection nearby. You don't want to kill the macrophage. It did its job.
You want to help it.
You want to activate the CD4 general to organize a larger response. Tell the B cells to make antibodies, supercharge other macrophages, call in more troops. The pathway determines the strategy.
That distinction, kill the infected cell versus help the messenger, is absolutely crucial. Now, Murray mentions there's one important exception to this rule called cross-presentation.
Ah, yes, the loophole. This is mainly a trick that dendritic cells can do, and it's vital for fighting certain viruses.
Give us the scenario.
Okay, imagine a virus that only infects kidney cells. It doesn't infect dendritic cells. The infected kidney cells will display viral peptides on their MHC-I, but they're stuck in the kidney. They can't travel to the lymph node to alert the army of naive CD8 T cells waiting there.
And the dendritic cell, which can travel to the lymph node, isn't infected, so it doesn't have any viral proteins inside it to load onto its own MHC-I. So you have a stalemate. How do you get the CD8 killers activated?
Cross-presentation is the answer. The dendritic cell goes to the kidney, eats a dying infected kidney cell. That's an exogenous antigen. Normally, we would put it on MHC class 2.
Yeah.
But the DC has a special pathway to shuttle some of that exogenous material over to the endogenous MHC-I pathway. It loads the viral peptide onto its own MHC class I molecule.
So it's presenting an external antigen as if it were internal.
It's cross-presenting it. This allows the dendritic cell to go to the lymph node and train a brand new CD8 sniper against a virus that the DC itself doesn't even have. It's absolutely critical for mounting a killer T-cell response against many viruses and tumors.
Okay, we've got the T-cell interacting with the APC via the TCR and the MHC. But Murray and Jowetz both stress that simply plugging that key into the lock isn't enough to start the engine. We need the two-signal hypothesis.
This is perhaps the most important safety mechanism to prevent autoimmunity in the periphery. Think of it like a nuclear launch sequence. You need two different keys turned simultaneously.
What's signal one?
Signal one is the specific recognition we've been talking about. The T-cell receptor binds to the specific MHC peptide complex. This ensures specificity. Your T-cell is now focused on the correct target.
But if that happens all by itself?
Nothing happens. Or even worse for the T-cell, it gets a signal to shut down permanently.
So you absolutely need signal two.
You need signal two. This is called co-stimulation. And the classic canonical co-stimulatory signal is a molecule called B7 on the APC.
Also known as CD80 or CD86.
Right. And it binds to its receptor on the T-cell, which is called CD28. When B7 plugs into CD28, that is the confirmed you have permission to fire signal.
So TCR binding is, I see the enemy. And CD28 binding is headquarters confirms this is actually a dangerous situation and not a false alarm.
That's a perfect way to put it, because professional APCs only put B7 on their surface when they sense danger. They need to be activated by the innate immune system first by seeing bacterial products via their toll-like receptors, for example.
So a T cell can't just accidentally activate against a healthy, uninflamed tissue cell that isn't showing any danger signals.
Correct. No danger means no B7, which means no signal 2.
Now here's the exam trap. There's another molecule on the T cell that also binds to B7, right? CTLA4.
Yes. This is the brake pedal. So after a T cell gets activated by B7-CD28, it does its job, but then after a few days, it starts to put a different receptor on its surface, CTLA4.
And what does that do?
CTLA-4 binds to B7 on the APC even stronger than CD28 does. It outcompetes it. But when it binds, it sends a powerful inhibitory signal into the T cell. It's the stand-down order. It turns the T cell off.
Why? To prevent the response from going on forever.
Exactly. You need to terminate the immune response once the threat is cleared. You don't want the army of T cells rampaging forever, causing collateral damage to healthy tissue. CTLA-4 is a crucial off switch.
And this is now a huge target in cancer therapy with checkpoint inhibitors.
It is. Drugs like ipilimumab are antibodies that block CTLA-4. They essentially cut the brake lines on the T cells.
Which allows the T cells to keep attacking the tumor because the off signal is being blocked.
Correct. We're manipulating this fundamental off switch to our advantage in fighting cancer.
Fascinating. But let's go back to that fail state. What happens if a T cell gets signal 1? It sees its antigen on an MHC, but there's no signal 2, no co-stimulation.
It enters a state of unresponsiveness called energy.
What does that mean functionally?
It means the T cell becomes a zombie. It's still alive, it's floating around, but it is functionally switched off. Even if it later encounters that same antigen with proper co-stimulation, it won't activate. It has been permanently tolerized. This is a crucial way the body maintains peripheral tolerance to harmless antigens like food proteins in the gut that shouldn't be triggering inflammation.
Okay, so once the T cell gets both signals, it finally wakes up. And Murray points out that the first thing it does is secure its own supply lines. The IL-2 autocrine loop?
Yes, this is its rocket fuel. The newly activated T cell starts furiously secreting a cytokine called interleukin-2, or IL-2. At the exact same time, it puts a high-affinity version of the IL-2 receptor, also known as CD25, all over its own surface.
So it's making its own growth factor and building the best possible antenna to receive it. It's self-sufficient.
It is. It's an autocrine loop. It feeds itself. And this IL-2 signal is the command to divide, divide, divide. This is what drives clonal expansion. That one single T cell that recognized the pathogen can become an army of 10,000 identical clones within a week.
So now we have an army of activated CD4 helper T cells. But they don't all do the same thing. They differentiate into specialists. This is where we meet the different generals of the battlefield, the T helper subsets. And honestly, reading this section, it feels like a geopolitical struggle. Th1 versus Th2.
It is absolutely a war within a war. And what determines which path a naive CD4 cell takes is the cytokine environment it's born into. What is the innate immune system screaming about at the time of activation?
Let's start with Th1. What's its specialty?
Th1 is the cell-mediated immunity specialist. It's induced to form by a cytokine called IL-12, which is primarily made by macrophages and dendritic cells that have eaten an intercellular pathogen.
So the innate system says we've got an inside job.
Exactly. The Th1 cell's main job is to produce a powerful cytokine called interferon gamma, or IFN gamma.
And I want to pull in the Jowet's perspective here because it clarifies the why of IFN gamma so well. T1 isn't just pro-inflammatory. It's specifically the rescue squad for incompetent macrophages.
That is a fantastic way to put it. Think about pathogens like tuberculosis or listeria. These are bacteria that have evolved to survive and even thrive inside the macrophage's lysosome. The macrophage eats them, but it can't digest them. It's been hijacked.
So the Th1 cell comes along.
It recognizes the bacterial peptide on the macrophage's MHCT2, and it sprays that macrophage with IFN gamma. This supercharges the macrophage. It dramatically ramps up its production of reactive oxygen species, nitric oxide, and lysosomal enzymes. It turns the macrophage from a comfy home into a blazing furnace.
So Th1 is all about dealing with intracellular threats. What about its rival, Th2?
Th2 is for the big stuff that you can't eat, the things that live outside of cells. The classic targets are parasites, especially Helminth's worms, and unfortunately for many of us, allergens.
What's the induction signal?
The Th2 differentiation is driven by a cytokine called IL-4. And once formed, the Th2 cell produces a whole suite of its own characteristic cytokines, IL-4, IL-5, and IL-10.
Break those down for us. What do they do?
IL-4 is the signal that tells B cells to class switch their antibodies to make IgE, which is the specialized anti-parasite and allergy antibody. IL-5 is the signal that recruits and activates eosinophils, which are specialized white blood cells that are very good at killing worms.
And IL-10?
IL-10 is an anti-inflammatory cytokine, and its main job is to actually suppress the Th1 response.
Wait, they actively fight each other?
Constantly. It's called cross-regulation. Th1 via IFN gamma actively inhibits the development of Th2 cells, and Th2 via IL-10 and IL-4 actively inhibits the Th1 response. It's a balance. The body has to commit to one strategy or the other.
And if it chooses the wrong strategy,
It can be fatal. The classic textbook example is leprosy.
which is caused by Mycobacterium leprae, another intracellular bacterium.
Right. And patients with leprosy present in two very different ways. In tuberculoid leprosy, the patient mounts a very strong Th1 response. They produce lots of IFN gamma. Their body is able to wall off the bacteria inside granulomas. The patient might lose some skin sensation, but they generally survive and the infection is contained.
But in the other form, lepromatous leprosy,
The patient has a defective Th1 response and mounts a very strong but completely inappropriate Th2 response. They make tons of antibodies, which are useless against an intracellular bacteria. They make no IFN gamma, and the bacteria just run rampant throughout their body. The patient suffers severe disfigurement and widespread tissue destruction. It's a catastrophic failure of choosing the right tool for the job.
this also ties into the hygiene hypothesis for allergies, doesn't it?
It does. The idea is that in our super clean modern world, we don't get enough early childhood infections that would normally train and strengthen our Th1 system, so our immune system defaults to being Th2 dominant.
And a bored Th2 system starts attacking harmless things like pollen and dust mites.
That's the theory. It leads to the epidemic of allergies and asthma we see in developed countries.
Let's briefly mention the other two major subsets, Th17 and TREGs.
Th17 is another pro-inflammatory lineage. It's critical for fighting extracellular bacteria and fungi, particularly at mucosal surfaces. It produces a cytokine called IL-17, which is a potent recruiter of neutrophils.
But when it's dysregulated,
It's a major player in many autoimmune diseases like rheumatoid arthritis, psoriasis, and inflammatory bowel disease. And then you have the polar opposite. The regulatory T cells, or TREGs,
the peacemakers.
they are the peacemakers. They are defined by a master transcription factor called FOXP3. Their job is to produce anti-inflammatory cytokines like TGF-beta and IL-10 to shut the whole immune response down when the infection is over and to maintain tolerance to self.
Okay, so those are the generals, the CD4 cells. Now let's talk about the assassins, the CD8 cytotoxic T cells. These guys are terrifyingly efficient killers.
Once they're activated, which often requires licensing help from a Th1 cell, they leave the lymph node and roam the tissues, systematically checking the MHC class I windows of every cell they meet.
And when they find their target, say, a flu-infected lung cell,
They engage in what's often called the kiss of death.
it sounds ominous.
It is for the target cell. The CD8 cell forms a very tight, organized connection with the target cell called an immunological synapse. Then it reorients its internal machinery and releases cytotoxic granules directly onto the target surface. These granules contain two main weapons, perforin and granzymes.
Perforin sounds pretty self-explanatory.
It perforates. It's a protein that polymerizes and punches holes in the target cell's membrane. This allows the second weapon, the granzymes, to get inside.
And what do granzymes do?
Granzymes are a family of enzymes. Once inside the target cell, their job is to activate a family of suicide enzymes within the target cell itself called caspases.
And caspases are the executioners that trigger apoptosis.
Correct. And the distinction here is absolutely vital and a common exam question. CD8 cells cause apoptosis, which is a clean, quiet, programmed cell suicide. The cell shrinks, its DNA gets chopped up, and it breaks into neat little packages that are cleaned up by macrophages.
They do not cause necrosis.
They do not cause necrosis, which is a messy, explosive cell death. Necrosis would spill viral particles everywhere and cause massive inflammation. Apoptosis is a clean, controlled demolition. You want to take out the virus factory without burning down the whole neighborhood.
And they have a backup weapon system, too.
They do. The Fas-Fas ligand pathway. The CD8 cell has a protein on its surface called Fas ligand. It can bind to a receptor called Fas on the surface of the target cell. This binding also directly triggers the caspase suicide pathway. It's a second, independent way to tell the cell to die.
Ruthless. And they're serial killers, right? One CD8 cell can kill multiple targets.
Oh, yes. After it delivers the death blow, it detaches, rearms, and moves on to the next infected cell.
Incredible. Okay, let's pivot now to the other major arm of the adaptive system, the B cells, the artillery division.
If T cells are all about close quarters, hand-to-hand combat, B cells are about long-range missile strikes. They secrete antibodies that travel through the blood and lymph to neutralize threats far away.
We mentioned earlier that B cells can be activated in two main ways, T-independent with that Velcro effect, and T-dependent. Let's focus on T-dependent because that's where all the sophisticated stuff happens.
All right. So a naive B cell finds an antigen that fits its B cell receptor, which is basically a surface-bound IgM molecule. It pulls the antigen inside via endocytosis, digests it, and presents a piece of it on its MHC class II molecule.
Wait, hold on. B cells are APCs. I thought that was just macrophages and dendritic cells.
Yes, that's a key point. B cells are excellent antigen-presenting cells, but they're very specific. A macrophage will eat anything. A B cell will only eat and present the specific antigen that its receptor recognizes.
So it's presenting what I caught to a helper T cell.
Exactly. It finds a specialized T helper cell in the lymph node called a T follicular helper cell, and this is where the critical handshake happens.
The second signal for the B cell.
It is. The T cell uses a molecule called CD40 ligand to bind to the CD40 receptor on the B cell. That interaction, plus cytokines from the T cell, is the magic signal. It tells the B cell, I confirm your target is legitimate. Proceed with activation and improvement.
And proceed means moving into the germinal centers of the lymph nodes. This is where the B cell gets its upgrades. First upgrade, somatic hypermutation.
This is a truly wild biological process. The activated B cell starts intentionally introducing random point mutations into the DNA that codes for the variable region of its antibody.
It's literally gambling to try and make a better receptor.
It is evolution on fast forward happening inside your lymph nodes. It's trying to improve the fit of the antibody for the antigen. This is a process called affinity maturation.
Getting the tighter grip.
Exactly. The B cells that make a mutation that binds weaker to the antigen don't get survival signals, and they die. The B cells that make a mutation that binds stronger get survival signals from other cells in the germinal center. So over a few weeks, you select for antibodies that go from a loose handshake to a death grip.
And the second upgrade is class switching.
Right. The B cell now changes the handle of the antibody, the constant or FC region, to change its function. The part that binds the antigen stays the same, but the tail changes. It starts by making IgM, but depending on the cytokine signals it gets from the T cell, it can physically loop out a piece of its DNA and switch to making IgG or IgA or IgE.
Let's run through those isotypes. Murray suggests the mnemonic GAMED for the 5 classes: IgG, IgA, IgA, IgE, IgD. Let's hit the key structure and function for each one.
Okay, let's start with IgM, the first responder.
It's huge. It's a pentamer.
5 Y-shaped antibody units all tied together. That gives it 10 antigen binding sites. Since the early immune response has antibodies with low affinity, a weak individual grip, the pentamer shape gives it very high avidity, a strong overall grip from having so many hands.
It's like a grappling hook.
It's a grappling hook. It's also the best isotype at activating the complement system because of its shape. But its huge size means it's mostly stuck in the bloodstream. It cannot cross the placenta.
Okay, next up, IgG.
IgD is the special ops antibody. It's a small monomer. It's the most abundant antibody in your blood. It can easily squeeze out into the tissues. Crucially, it is the only isotype that can cross the placenta, which provides the baby with mom's immunity for the first few months of life.
And its main functions.
It's the best opsonin. It coats bacteria to make them tasty for phagocytes. And it has the longest half-life, about 23 days. This is your main memory antibody.
What about IgA?
IgA is the border patrol. It's found mainly in secretions, in your gut, your lungs, your tears, your saliva, and in breast milk. In those secretions, it exists as a dimer, 2 units held together by a J chain, and it's wrapped in a protective protein called the secretory component.
Why does it need the secretory component?
It acts like an armored vest. It protects the IgA from being digested by all the enzymes in your gut and other mucosal surfaces. Its job is to neutralize pathogens right at the entry points of the body. IgE is the grenade. It's found in very tiny amounts in the blood because most of it is stuck to the surface of mast cells and basophils. If it then encounters its specific target, a parasite or an allergen like pollen, it triggers that mast cell to degranulate, to explode with histamine.
That's the basis of type I hypersensitivity or allergic reactions.
Exact.
And finally, IgD.
IgD is the mystery isotype. It's mostly found on the surface of naive B cells right alongside IgM. Its main role seems to be in signaling for B cell activation, but you rarely find it secreted in significant amounts in the blood. Its function is still a bit murky.
Okay, Murray puts all of this together in a timeline that is really, really important for diagnosing patients in the clinic. The primary versus the secondary immune response.
This is the entire logic of serology testing. When you are first exposed to a new antigen, the primary response, There's a lag phase. For maybe 5 to 7 days, nothing seems to be happening while your adaptive system gets going.
The innate system is holding the line.
Right. Then, the first antibody you see rise in the blood is IgM. It spikes fairly quickly. Then, a little later and more slowly, you see the IgG levels start to rise as class switching occurs.
So if I draw a patient's blood and I see high levels of IgM specific to a virus and little to no IgG.
You are looking at an acute, active, primary infection. It's happening right now.
But what if that same person gets re-exposed to that virus years later?
That's the secondary or anamnestic response, and it is completely different. There is almost no lag phase, maybe a day or two. The army of long-lived memory B cells wakes up almost instantly and starts pumping out massive amounts of antibody.
And it's not IgM this time.
No. The dominant antibody is IgG right from the start, and the IgG titer goes much, much higher than it did in the primary response. Plus, due to affinity maturation, it's a much higher quality, tighter binding antibody.
So if I see high levels of IgG in a patient's blood and no IgM,
that indicates a past infection or that they've been successfully vaccinated. They are immune.
And there's a classic clinical trap involving newborns.
Yeah. Yes, this is a very high yield point. A newborn baby has IgG in its blood. Is it infected? No. That is mom's IgG. It crossed the placenta. This is passive immunity, and it protects the baby for about 6 months until its own immune system matures.
But if a newborn has IgM,
IgM is a giant pentamer. It is too big to cross the placenta.
so if the baby has IgM, the baby must have made it itself.
Exactly. Finding IgM specific to a pathogen in a newborn's blood means the baby was infected in utero. It is diagnostic for congenital infections like the Torsades infections, toxoplasmosis, rubella, CMV, and so on.
That is the kind of detective work that makes immunology so cool.
It really is beautiful logic. You know, adaptive immunity is expensive. It takes time to get started, but once it learns, it provides a level of precision and memory that is absolutely essential for keeping us alive in a world full of evolving pathogens.
All right, speaking of precision, I think it's time to test our listeners' memory. We're going to do a rapid-fire recall session. I'm going to give you a clinical scenario based on what we just discussed. You at home, you have a few seconds to think, and then our expert will drop the answer and the why.
Let's do it. Review mode activated.
Scenario 1. You have a pediatric patient with recurrent candid infections and autoimmune problems like hypoparathyroidism and adrenal insufficiency. Genetic testing reveals a mutation in the AIR gene. What specific part of T-cell development has failed in this child? Pause.
The answer is negative selection, which is the main mechanism of central tolerance. Without a functional AIR gene, the thymus cannot display those peripheral self-antigens. Therefore, T cells that are strongly reactive to self are not deleted. They survive, graduate from the thymus, and go on to enact the body's own tissues.
Scenario 2. A patient is vaccinated with a pure polysaccharide vaccine, so there is no protein conjugate, which antibody isotype will be predominantly produced, and importantly, will they develop long-lived memory B cells? Pause.
The answer is they will produce IgM only, and they will not develop memory. Polysaccharides are classic T-independent antigens. They can cross-link B-cell receptors to get a quick initial response, but they cannot recruit T-cell help. Without T-cell help, specifically the CD40L signal, you cannot get class switch recombination to IgG, and you cannot form memory B cells.
Scenario 3. A patient receives a kidney transplant from an unrelated donor. A week later, the graft is showing signs of acute rejection, mediated by the patient's CD8 plus T cells. Which specific molecular structure on the donor kidney cells are the host CD8 cells recognizing as foreign? Pause.
The host CD8 cells are recognizing the donor's MHC class I molecules. Remember the rule. CD8 cells interact with MHC class I. All nucleated cells, including the cells of the donor kidney, express class I. The host T cells see the donor's specific MHC molecules, their HLA type, as foreign, and they attack and kill those cells.
Scenario 4. A new immunosuppressive drug is developed that works by blocking the interaction between B7 on the antigen-presenting cell and CD28 on the T cell. The T cell's receptor still binds its target antigen MHC complex, but this drug is present. What is the functional outcome for that T cell? Pause.
The outcome is energy. The T cell becomes functionally unresponsive. Remember the two-signal hypothesis. Signal 1, which is the antigen binding, without signal 2, which is the B7-CD28 co-stimulation, does not lead to activation. Instead, it leads to a long-term state of unresponsiveness or anergy.
Okay, final question. Scenario 5. A child presents with a parasitic worm infection. You would expect their lab work to show elevated levels of IgE. Which specific cytokine is required to tell a B cell to class switch to produce IgE, and which TL per subset is the primary producer of that cytokine? Pause.
The cytokine is interleukin-4, or IL-4, and it is produced by Th2 cells. The Th2 lineage is the specialized anti-parasite and allergy arm of the helper T cell response. IL-4 is the specific polarizing cytokine that tells B cells to switch their heavy-chain constant region gene to the epsilon class, which codes for IgE.
How did you do out there? If any of those tripped you up, I really recommend checking out the specific sections in Murray Chapter 9.
And honestly, look at the diagrams. The textbook has fantastic figures that visualize things like the VDJ recombination loops and the MHC grooves. Seeing it visually makes this stick so much better than just listening.
Adaptive immunity is really the story of how our bodies learn from our environment. It's what keeps us safe from the things that tried to kill us once, ensuring they almost never get a second chance.
It is the ultimate biological learning machine.
Thanks for diving deep with us. We'll see you in the next deep dive where things get even smaller and somehow even more complex. We're heading into the world of virology. Stay curious.
Stay immune.